I'm a disease ecologist, and as a disease ecologist, my perspective on disease is that it is itself an emergent property of a very complex system. And this is an image you might have seen before. It was published in the New York Times many months ago, years ago. I think it's COVID time warp, so everything's blurring together. Remember the good old days when disease ecologists were not front and center? But this is more of what we're seeing now. We have to understand the basic ecological interactions that are underpinning these emergence events. When we talk about diseases that persist in wild animals and spill over to infect humans, these are called zoonotic diseases, and they are there whether or not humans are involved in the picture. They persist without impacting the organismal populations, and they're there in the wild with no regard for humans. And the reason why they spill over is because of the changes that we impose on the system. And so we've heard throughout the last couple of days, some of these complex systems that are contributing to these emergence events. And that includes things like climate, things like water. We've heard about food security. Animals suffer also from food insecurity. When we change the landscape, we change their access to limited resources during stressful periods of their life cycle, and that has impacts for disease emergence as well. So there's a lot of complex things happening here, and I think the way forward must be an interdisciplinary perspective. And so that has been the approach that my group has been taking. What we do often is place this complexity, this ecological complexity, on the back burner, especially when we have an outbreak happen. When an outbreak happens that grows quite large, we get afraid and we try to put out that fire as quickly as possible. And so what we end up with is a system that is sort of heavily driven by an emergency context. We're constantly moving quickly to go from fire to fire. And when we get a break between those diseases, like the break between the 2015 Ebola outbreak and the next Zika outbreak, we transition for a bit into this peacetime warning phase where we think about intervention, we think about how to make sure that doesn't happen again. We tend to study diseases that have been given names, that have diagnostics associated with us being able to find them. And so we study things that we can see because they have already emerged. But by then, the risk has already been realized. The risk that comes from this watch zone has already appeared on the landscape. And so we're already behind the curve. So what my research program aims to do is try to take the data that we have to work with right now and move that effort upstream so that we are not waiting in the warning phases and in the emergency phases, constantly fighting fires. And we can start to imagine a world where we're managing things well enough that we don't have to constantly be firefighters. And instead we can invest more in understanding the knowledge upstream that is really underpinning all of these interactions and therefore disease emergence. So I'm going to give two quick examples from recent work from my group to highlight how we try to approach these problems. And the first example is from Ebola virus, which all of us are pretty familiar with. And I think the thing that still strikes me about this system is how little we still know about the system. It's been around for 40 years. And while a lot of the evidence points to bats being a major player in the reservoir game, we still don't have evidence of replicating virus in a single animal. So despite 30, 40, 50,000 samples from hundreds and hundreds of species across Africa and other places, we still haven't found an animal that has live replicating virus in its body, which is quite remarkable because this has been breaking out and spilling over for decades now. And as you all know, we have an active outbreak in Uganda at the moment. So the question marks here are also quite striking to me because while Ebola has been found in many dead animals, it does kill quite a lot of hosts. Its reservoir, the way that it transmits from the reservoir to these animals is also still quite unknown. So to try to get a handle on this, or to try to understand this better, it helps to understand the context of the problem, right? There's a lot of species. Bats can travel quite far, and bats are also very species rich, meaning there's lots and lots of them. 1,116 species, I think, is the most recent count. And that is just right behind the rodents, which is at 2,277 species. So those two groups alone comprise the majority of mammals, which is about 5,000 species. And if this area is quite striking, right? Like you can fit the US, China, and India within the area of Africa. So, and this area is not just large, but it's incredibly biodiverse. It's mind-bogglingly diverse. And so to think about trying to find a host species at the moment that it's shedding virus in an area this size, when there's thousands of species that are moving in time and space, is a Herculean task. It's just hard to do. And field biology is not easy. Catching bats is definitely not easy. So these red areas represent places where the outbreaks have occurred in the past and where they're continuing to occur now. So let's take a slightly different view of this picture. If we take all of the bats that have ever tested positive, and I use my quotation fingers because tested positive just means that we have evidence that they've been exposed. Their bodies have a reaction to having been exposed to the virus, and you can detect that through serology, through their serum. It doesn't mean that they're actively infected. It just gives you a signal that Ebola is there. So if we gather that scant evidence together, there are just 21 species at the time of this publication for positive bats. And that kind of splits evenly between fruit bats and insectivorous bats. Fruit bats are the big ones. Insectivorous ones tend to be kind of small. And here's where they're distributed. Now, we generated this map after the 2015 outbreak, which was centered right here, okay? So had we made this map before 2015, it probably would have been a no-brainer to say we should maybe keep an eye on the red zones where lots of species are overlapping in space, because there's an indication that the risk might be higher there. And that's about the best we could do. Overlap the geographic ranges and see where they're overlapping. And then you can see the other red zone is here, right where we might expect it to be given the history of spillovers. Now, what other species, these are just 21, and there's thousands of other ones here. Which ones should we be sampling if we're looking for that reservoir? This is the question that we set out to answer when this outbreak began. We had developed tools that drew from species level traits that help us to distinguish one species from another. And these are intrinsic differences. So they're not traits that change quickly in response to weather or in response to like where they are within their range. These are traits that collectively give us an evolutionary snapshot of how these species have evolved their life history strategies to contend with multiple selection pressures over millions of years. And those selection pressures include things like the environments that they prefer, places that they're adapted to, but also parasites and pathogen threats that they have overcome and have now been persisting for maybe thousands of years in an equilibrium to where the pathogen is not overtaking them. They are persisting in equilibrium with this pathogen, or at least able to outrun it. So these traits are really important for helping us to understand the capacity of a species for serving as this kind of a zoonotic reservoir that we're interested in detecting. And so the challenge here for the machine learning is whether we can train an algorithm to recognize the characteristics of a species that should serve as a reservoir, even if we don't know that that thing is a reservoir. And even when we only have a few data points to go from. So in this case, it's not like probably IBM has to do with much more sparse data, but this is, we have like 21 out of 1,100 species that we're working with here. So it was a pretty difficult training task, but the upshot is that it worked. So I skipped over all of the math and all of the model results. I'll just say for the computer scientists in the room that we used a generalized boosted regression model and 80-20 split, 10-fold cross-validation, all that stuff. But essentially the take home is that it did work. The model predicted filovirus positive bats, so anything that was infected with Ebola, Marburg, and all the related viruses, with about 87% accuracy after correction. And it did it on just the basis of those traits, just the traits. No information about molecular compatibility, no serology, nothing else about the immunology, just their traits. So how do we know that this worked, right? Well, we can wait for passive validation is generally the way it's done. There's no completing of the loop, right? So if we have a model, we make predictions. The next thing that makes sense is to take those predictions and test them, right? Test them in the lab, go out and target those species in the field. Well, we do not have a mechanism of completing that loop right now in science. We do it haphazardly and when the funding is available. And because we have this panic neglect cycle in disease response, often once these kinds of results come public, the money's moved on to Zika and there's nobody interested in Ebola anymore. I think that's why these networks are super important to keep this kind of work going. So the passive validation did come, unfortunately. We had the dubious honor of being right on multiple occasions. So this guy is in red. These are epauletted fruit bats. And this one's in red because it was already known to be positive. And our model predicted this one as one of the top predicted species, very closely related. They'd have distinct geographic ranges, but they're very closely related. I think that there's just one ridge on the rooftop of the mouth that distinguishes one species from another. They also look a little different, but we're not really focused on looks here. Here at the bottom, these two species are really interesting. This is the Angolan free-tailed bat, and this is the little free-tailed bat. And these guys have a very broad distribution, including right here in Kenya. And I think these species were also sampled in Sierra Leone. And I bet there's a few other publications showing that they're positive for a new filovirus that was just discovered a few years ago in 2019. It's been given the name Bombali virus. This is a virus that has since been experimentally shown to be able to enter human cells, which is an indication that it does have zoonotic potential for humans. I'm not sure whether the research stands in terms of being able to replicate in the cell. There's all these other things that have to happen once that virus gets into the cell in order for the reservoir to actually serve as a true reservoir. But these guys were collected from roosts that were in people's homes. So the transmission possibility is there, the contact interface is there, and the prediction is there. The question is, where do we take it next? And to my knowledge, it's not moved any further than this. Here's another species. If we look at this map, you see that there's some action going on here in Southeast Asia. Let's go to Southeast Asia. There's a bunch of new filoviruses always being discovered in this region. However, as we all know, there's not been an outbreak of Ebola or Marburg or Reston or anything like that in this region. And the question is, why not? What's different about this region and these species and these ecosystems that are preventing or, they're not conducive to the kinds of spillovers that we're seeing regularly in Africa? This is a burning question for me, and I don't know how to think about this other than there's something biologically different about the species here, and there might be some background resistance to the populations in this area that are preventing the spillovers from happening. This was another example of a species that was passively validated about 6 months after we published our paper. Obviously, these guys were doing the field work while we were doing our modeling, but they found this is a new host for a new filovirus in a new area of the body. So it's now infecting the lungs. They found it in the lungs, which suggests that there might be an aerosolizable route of transmission. And it was number 5 on our prediction list. So that's what the models were able to do. They can give us a picture of what traits are important for getting that accuracy high. They rank order the species according to which ones have the traits that make it seem like it should be a candidate for targeted surveillance. But of course, this kind of model is only possible when you have data. We had not a lot of data, but we had some data to go on. What happens when you don't have data? And that's exactly the situation we found ourselves in when this happened. So now with the benefit of hindsight, we know that animals can get infected by humans with COVID. They can die from COVID, and some of them can get COVID and transmit COVID back into humans. So there is this spillover, spillback, and secondary spillover events happening all the time now in nature. And the question is, how do we predict which species to focus on? And how do we know if that matters for us? The most recent studies show that alpha and delta variants are stable in white-tailed deer, which means that they're not really evolving. But we know that our immunity wanes and the variant landscape is always changing. So what does that mean in the future? Are we going to be able to get it back from white-tailed deer, which has been already shown, by the way? But look 10 years down the line, how do we know what's going to happen? These are the questions that I'm super interested in pursuing next. So what we did here, because we had no data and because we had very few species that had been tested positive at the beginning of the pandemic, we had to kind of go back to first principles. And we asked ourselves, well, in order for infection to occur, it doesn't matter what direction you're thinking about here. There's one thing you have to do as a virus. You have to be able to bind that cell receptor. If you can't bind it, you're not going to get in and nothing else is going to happen. So let's go back to the basics and see how many species have ACE2. And this was a situation where oddly we had too much data because ACE2 is found in all vertebrates. Okay, that's not really helpful. I'm not going to go and sample fish and birds. And but what we started with was we took this binding and said, we can estimate the strength of this binding using a molecular structural model. So if you train a model to take into account all the strong and weak intermolecular forces that are controlling this binding strength, you can rank order that where the higher binding is at the top. And you can make a sort of arbitrary cutoff at cats, domestic cats. They're the species that has the weakest binding, but that still gives you a productive infection. So theoretically, infection transmission is possible. So we just drew a conservative cutoff here. So everything that's above this line is like maybe transmissible infection. So we binarize the data in this way. And instead of looking at like fish, amphibians, birds, cartilaginous fishes, mammals, and reptiles, we just looked at the mammals because we thought those were the ones that mattered the most. So let's not get too caught up in the details here. I'm just going to walk you through what we did. We collected all of the evidence that was available at the time for any animal that had been confirmed positive for COVID or SARS-CoV-2. We then said, okay, well, we have to model the binding strength. Let's do that first. We take the binding strength scores and say the structure of ACE2, the reason why all the vertebrates have it is because it's so fundamental to controlling things like homeostasis, blood pressure, and osmotic balance. These are things that are super important for organisms successfully undertaking metabolism and dealing with their environmental constraints. And so that's why they have it. And the structure of molecules recapitulates their function. So small changes in these structures have meaningful consequences for how they act in the body. So we reasoned that if that is true, then it must have a concrete link to their traits, these intrinsic traits that separate species from each other. So using that logic, we said, all right, we're going to train a machine learning model to see if it can predict binding strength, thresholded binding strength with the trait data. And then we get sort of an estimate for which species have the capacity to transmit SARS-CoV-2 forward, okay, either to another species or to humans. And then we get a list of predictions, and ideally, we compare it. We did compare it to the confirmed hosts and then hosts that were predicted by other models to see how good of a fit it was. But really what we want to do, again, is complete this loop, which we still have not done. The next step here, I think, is to do the cell entry assays to see if the variants can actually get into the cells that we think it can, and then do the really sort of purposefully interdisciplinary studies, not just me publishing my thing and then waiting for somebody else to do their thing and then somebody else coming along and going, let's write a synthesis paper. And I think that's too slow. Like Charity was saying, how do we 10x this process? We need this loop to be normal, not special. So what did we find? A lot of things that you probably not surprising. Primates, very closely related to humans. Many of them, actually most of them, have the capacity to transmit. There are a bunch of rodents, a bunch of carnivores, bats that, you know, that's not surprising because the rhinolophids were the ones that have the other coronaviruses that are very closely related to SARS-CoV-2. And then a couple of other interesting species that have made the news because of the types of coronaviruses that they carry. But I'm just going to cherry pick a few species that were at the top of our prediction list from the continent. And some of these are Kenyan species, but these are not uncommon. Some of them are not like in your backyard, but they are species that that we do see that we're familiar with and that haven't been tested to our knowledge. There's no information about samples being collected from these species. Actually, maybe Mercy, you can correct me if I'm wrong. So these are some species from Africa. And, you know, we're constantly monitoring these other conformations. So when we published our paper, a few months later, well, we had, you know, we just sat back and watched like we always have to do, which is so depressing. He published this list and you're like, I hope you're wrong. I hope the model doesn't work. And then the snow leopard died in the zoo. That was very top of our list. White-tailed deer was confirmed, red fox. The raccoon dog was found to have a transmissible infection. Deer mice, which is very common in the US, positive for infection and can also transmit it from mouse to mouse when they're not touching each other. So in a cage with separation. And the deer mouse is super closely related to this white-footed mouse, which gets into my house. It's so annoying. I caught this guy on my countertop in my kitchen. I live in New York, so it's not uncommon, but the contact, this is not socially distanced. The contact is there. And then there's some, but it hasn't been shown to be infected. So there's a big question mark. And then cattle and pigs, there's some mixed evidence from cattle and pigs. So I'm going to wrap it up. I'm going to skip over these and just tell you that This complexity is actually critical. We have to grapple with this in an interdisciplinary way. And I don't know if the artist meant to do this, but I noticed that the background here is all blue. Blue sky research is what we need. This basic science. If we don't understand the basis of why these organisms behave the way they do and function the way they do, then how can we hope to understand emergence? These diseases are going to keep coming, and I haven't even talked about bacteria yet, but thankfully, Dr. Dushan Malloy is going to talk all about another threat, antimicrobial resistance, which is going to increase because of the kinds of therapeutics that we're throwing at these emerging pathogens. And of course, once the pathogens do emerge, we have these incredibly well-connected communities across the globe. And how can we're going to have to turn to these leading edge and creative data utilization methods to help us understand how transmission is going to happen so that we can figure out how to mitigate that. And Dr. Amy Wesolowski is going to talk to us about that. And just because we have all these new things emerging, we can't take our eye off the ball of these existing very perennial threats. And Dr. Esther Budajaya is going to talk about how her therapeutics uptake methods have been working for us in the tuberculosis world. Thank you so much for your time. I hope I've been able to convince you about the importance of basic research, and I'll turn it over to the next speakers.